So next one. So structure features. Anti-parallel strands. There are two strands over here running in opposite directions, 5' to 3', 3' to 5'. Simple. This mark indicates prime. 3' to 5', 5' to 3'. Clear? Next one. Sugar backbone. The deoxy has a sugar phosphate backbone. Then base pairing, nitrogen base pair, adenosine, guanosine, and chemical shape. And contribution of Chargaff's rules. So what is this Chargaff rule states? Chargaff rule states that the amount of adenine equal to tyrosine, A equal to T, and the amount of G is equal to C. Okay, this discovery was pivotal in understanding the complementary base pairing and ensuring the accurate application. So significance of base pairing. What is the significance of base pairing? Okay, any basis pairing. They will give replication fidelity. That means base pairing allows precise copying of genetic material, transcription accuracy, transcription and translation accuracies, and error prevention. So of course these things are mandatory to get a good replication process. So that's it. And that's it. So now tell me students, without seeing this data here, is it possible to find it from you? Yeah. What is the backbone of DNA? Phosphate and sugar, right? What are the strands? How many strands we have? Two anti-parallel strands. How they are running? 5' to 3', 3' to 5'. And what are the base pairing? What base pairs we have? And what are those bonds? Types of bonds between them? GC. Very good. And what is the diameter? Do you remember? Diameter of DNA? 20 nanometers. Very good. And helical turns. How many types of helical turns we have in DNA? Grooves. How many grooves we have? What are those? Minor groove and major groove. And helical turn. Each helical turn, from this side to this side, it consists of around 10 base pairs. So 10 base pairs per turn of the helix. So keep this in mind, okay? 10 base pairs are there for each helix. Yeah. Very good. Then, so see here, next question also very similar question. Discuss the complementary base pairing mechanism in DNA and its importance. I hope you already understood what is the meaning of complementary base pairing. A's complementary will be T, G's will be C. Okay? Yeah. So it says that complementary base pairing ensures accuracy, stability, or same thing. And mechanism of base pairing. Obviously there is a hydrogen bonding, the bond, and there is an anti-parallel orientation. If this sugar was this side, the other sugar will be downside. Anti-parallel, right? 3' to 5', 5' to 3'. So we have anti-parallel orientation. Then template function. DNA replication, each strand has a template for synthesizing the complementary strand. So importance of complementary base pairing, preservation of the genetic code, error detection and repairing, and transcription accuracy. That we'll see in the future. Actually, I started structure of DNA to discuss here actually. I thought of telling translation in the beginning, if you remember. That I will discuss later. Class is only till 3:30, right? Or 3:40? I hope you know, right? Amino acid is made up of 3 codons. Triplet codon. Have you heard of triplet codon? Triplet codon, the rich codon. What is your full name? Adenine. You are? Huh? Ashley. Okay. Adenine. Then in the next class I will discuss what is the translation. Okay, after translation I will discuss about the replication of DNA. Replication of DNA both in prokaryotes and eukaryotes. That's it. Any doubts so far? See, the subject is very easy. You need not to worry about much. You focus on your thesis and your synopsis. Because next semester, do you have research methodology in this semester? You study. So have you started working on your project? Writing paper?
We don't have.
You don't have a project. But anyway, start writing a thesis, okay? Start writing your, identify your research area. Because it will take almost 1 year to get published. Your seniors, your super seniors still didn't get their degree because they didn't publish their papers. Until and unless you didn't get your research publication, you won't get your master's degree in this university. And getting a publication is a very tough task. They all started in the end of their masters. And the problem is, in next semester you won't be in the campus. And you can't do these things alone unless and until we are guiding you. And if you are trying to come to campus, I will be engaged in classroom like this with someone else. So we won't be available easily to you. So the best time to do research is when you are present in the campus. And the second best time is right now. Start identifying your area and start working on it.
So what do we have to write on that?
Paper.
Yeah.
You need to do a project.
Proper what we got or what we go.
So you study research methodology, you might have some idea, right? Simple.
But we will only formulas and all that in research methodology.
I guess this is asking, you said we should know areas of research. So we find the areas ourselves, or we find them.
Yes, you need to identify your own areas. Once you identify your areas, then you will decide whether it's possible or not. If not, each faculty has their own expertise. Like I was expert in TB, and other persons are expert in biochemistry, Microbiology, Immunology. So you identify your area of interest, then we will see accordingly. What is your favorite subject? What is your name? Syed, right?
Yes, sir.
Yes, what is your favorite subject?
Microbiology, I like beyond from other subjects.
Microbiology is not a right word, that's a very broad statement. In micro, what is your favorite domain? Is it parasitology, biology, mycology, bacteriology? That you need to finalize. First, you are clueless where you are good at. Got it? So first identify your
Proper interest.
Proper interest where you are good at. Actually, for me, I like immunology a lot. So I went to ICMR at NIRP, National Institute of Tuberculosis Research Center. In that TB center also there is a department of immunology. So I went to department of immunology and I started working on TB. So in immunology, they are understanding that vitamin D has some immunomodulatory effects. Immune system was affecting macrophage digesting TB in the presence of vitamin D. Now I became a problematic situation. I said I love immunology, but my major domain became a microbiology because of TB, and my research area became a vitamin D, which is biochemistry. Now I need to understand the vitamin D metabolism. Then I got a real challenge. The real challenge is this vitamin D inactive form to active form is called 1,25-dihydroxycholecalciferol or calcitriol. Inactive form is one there. This conversion mediated by a enzyme called 1-alpha hydroxylase. Now this enzyme is coded by a gene. That gene is CYP27D1. In India, there are many people have vitamin D deficiency. I thought that this gene has some problem. This gene was inactivating the enzyme, leading to not converting inactive to active form of vitamin D. Only once it is activated, it will initiate macrophage to digest the TB. So now I started understanding the molecular biology of my gene. Then I came to know that there are 9 variations in this gene, single nucleotide polymorphisms in this gene. And I don't know which polymorphism is dominant in Indian population. And that is a wild research, no one touching these areas. And each polymorphism, I worked on RS1920401, which is restriction site single nucleotide polymorphism on one particular area of the gene. And the primer costed me around 1,20,000. Just primer. Without that primer, I cannot extend that portion of DNA in the PCR. And all primers are from US, they're in dollars. But thankfully, since I did my research in NIRT, the government funding and its ongoing mega project for them. So I did my 25 lakhs project free of cost because, I had a good grip in basics. So now what I'm trying to say is, unless and until you have good grip in your theoretical knowledge, you can't handle the research. Research is very tough. I was there just telling Vamsi there are 3 billion base pair bank. They say Vamsi you go to SNP bank and you identify what is your favorite gene, favorite mutation. How can I know? I need to go with literature. Some literature saying this gene is responsible for your cancer, some literature saying neuro problems, and some literature also saying that it can be affected VRPP by macrophage. Because that is what research means, it's unknown. And we need to connect the points. And my most painful problem was, after I said they requested, they brought the reagent. Once I started working on it, I am unable to get my strands and my DNA was not amplifying. And they have a PCR engine that is costliest PCR machine, not both. But my plan was that it is only running one sample but not multiple temperatures at the same time. Actually, each primer has its own melting temperature, but that can handle 19 different temperatures, but that machine not working on that particular type. If that worked, I might get results in 1 week, but it took me 7 months to get my. It took almost 2 months to get my amplified DNA, and they started doubting my capability. That scientists they are thinking that I am doing error in some kind of PCR validation, master preparation. You see, you are doing something wrong. Even I was not sure, is it my technical problem or is it my gene melting temperatures problem or primer problem. I started working on, I was calculating my melting temperature, K temperature. There are a few formulas, but I am going by temperatures are getting. But I came to a range. In that range, each day I'm keeping 67 one day, 67.5 one day, 70, 72.5, 72.7. At one thankful day, I got my extracted DNA visible in my gel. It took a long time. Yeah, that is what research means. You are really in a very good comfort zone. You don't know what you're missing. So start understanding research and identify your area. Sir, I thought if I fail in India, I will get my degree. Not demotivating us. That's it. Is there any class after this? Oh. Whose class is that? Practical. Okay.